opportunity. I would like to begin with thanking Professor Choro Che for inviting me to this wonderful place and giving me an opportunity to talk about what I have been doing. And I will talk about efficient nanotransporters, such as RRE, any experiment. So a little bit of theoretical work as an experimental chemist did, so very rudimentary. So probably I'll talk about theoretical result a little bit to satisfy some of your theoreticians. Okay, so I have no story tonight. Is about NO story, nitrogen monoxide. So as you know, nitrogen monoxide is a poisonous gas, and it's an also important intermediate in the chemical industry. And also, nitrogen monoxide is an important cellular signaling molecule involved in many physiological and pathological processes. For example, nitroglycerin, which is very old explosive and less known, is also heart medicine for atherosclerosis and the myocardial infarction patient. It's very hard English name, but it's something related to the heart problem. It is because nitroglycerin provide NO that increases blood flow by dilating the vessels. Interestingly, Alfred Nobel suffered from angina pectoris and was prescribed nitroglycerin, and it is another heart problems. So he thought it was really the irony of fate. He wrote a letter to his friend saying he was prescribed nitroglycerin internally. Okay. In fact, the nitroglycerin is the drug still used for heart patient, and the other form of nitrate drug is being synthesized, is being really actively being used. It's because NO is has many functional roles in our body, and name a few. Let's see. Name a few. It works as a neurotransmitter or immune system. Also, as I shown you, it works as it controls blood flow, and NO stays in many molecular forms in our body. And in fact, NO, the function of NO is still being discovered, and I cannot really follow what is really going on in this NO chemistry, because it is still really active area of research. But what I really got interested in about NO was about NO in cancer biology. It was reported that NO plays really key roles in cancer biology. Interestingly, NO can promote tumor cell growth, or at the same time, it can suppress the growth of the tumor cell. So clearly, the concentration of NO is really important if you want to do something about cancer cells with the NO molecule. And another thing you have to remember is NO has a very short lifetime, in particular in oxygenated environment. So to use this NO molecule, you like to approach using NO precursors. So NO precursor having very stable form, and you need a mean to release NO at some particular time with a very controlled concentration. So in other words, controlling the concentration of NO at a particular site at a well-determined time is pretty critical. In that sense, the photochemical strategy using NO precursor is very advantageous. So in that direction, you like to have a good NO precursor, which is thermally stable, non-toxic, water-soluble will be preferable. At the same time, you like to have a very detailed photodissociation dynamics of NO. As a physical chemist, this is the place where I can get into getting a detailed dynamics of NO. So I studied photodissociation dynamics of NO from Russin's red ester, which is RRE, using femtosecond time-resolved infrared spectroscopy. RRE is basically consists of 2 iron atoms and 2 thiol molecule and 4 nitrogen molecules. Basically, this alkyl substitute is only place you can play with. By changing this alkyl substitute, you can adjust the stability of this molecule, toxicity, or solubility of them. So simple way of approaching to get a water-soluble, low toxicity RRE was using amino acid cysteine or cysteine derivatives such as N-acetylcysteine called NOC or N-acetylpenisylamine called NAB. And the synthesis of RRE was simple enough for physical chemist can do by just mixing appropriate amount of iron atom and thiol molecule. We could get a cis RRE and nag RRE and NAB RRE. And we found out these molecules are thermally stable enough. We could do the photodissociation dynamics. So once we synthesize this stable RRE, we put it in the sample and prepare very intense femtosecond UV or visible pulse. Also, we prepare femtosecond mid-infrared pulse. And so starting from very stable RRE, and shine the intense light pulse, initiate the photoreaction, and using femtosecond IR pulse, you measure the infrared spectrum. And by adjusting optical delay and replacing a sample with a new compound, new sample, we can get another spectrum at a later time. You can repeat this experiment at various optical delay and can get a time-resolved infrared spectra of cis-RRE after excitation at 400 nanometer at the time 300 femtosecond after excitation to the 10 microseconds. So that was the broad time range. This is contour map of time-resolved infrared spectrum. And so this time span is long enough, we can cover entire reaction, starting from primary reaction to the secondary reaction, all the way to the end product. And at equilibrium spectrum, you can see a strong two band around 1800 wave number, which arise from NO stretching mode, anti-symmetric and symmetric. And also excite the molecule, some of the molecule undergoes reactions. Therefore, the intensity of the absorption band decrease a little bit. And so that shows as a negative signal we call the bleach. And that bleach signal shows up in negative color here. So you can see the negative color appears immediately, and that amplitude decreases slowly. And also you can see the red feature here, which is a new absorption band immediately appearing, and some of them appears later and decays. So there's a new species or new state comes. And so all these spectral features, spectral evolution can be reproduced using 4 basis spectra. Namely, one of them is RRE in the ground state. Another one is RRE in the low-lying electronically excited state. Another one is spectrum from RRE radical, which is deficient of one NO. And another spectrum is solvent-bound RRE. So by adjusting all this amplitude of these 4 spectrums, you can reproduce all the spectral features of the data. In the process of this feeding, in feeding, you can see the amplitude, how individual spectrum, time-resolved spectrum is being reproduced by composition of this base spectrum by adjusting amplitude. And each time we can plot this time-dependent amplitude of each basis spectrum, and you can see the basis spectrum has a certain nice timing behavior. And you can repeat this experiment by pumping several wavelengths, not just 400 nanometer, UV pulse like 267 nanometer, or 300, or 360, and various wavelengths. We found similar behavior, same basis spectrum. We could reproduce the basis spectrum and get a kinetics of it, and then summarize the photolysis kinetics of cis-RRE in water. So when we excite this molecule, cis-RRE, with a 260 to 400 nanometer pulse, that immediately go to the intermediate state, more than 50% go to the low-lying electronically excited state, which decays to the ground state with about 6 nanosecond time scale. So turns out to be this is just the simple photophysics, just going into the ground state, which we don't really want. But luckily, about 40 to 50% go to the photodissociation, generating radical and free NO. But this reaction is happening in solution phase. So this free NO and radical is surrounded by the solvent. So they are very nearby, so they can react immediately and undergo geometry recombination and go to the ground state. So that's not good. But some of the molecules happen to react with a solvent and forms a solvent-bound radical and leaving free NO. So this free NO can react with the target molecule. So that molecule is useful for delivery. It is useful for delivered molecule. So in here, still, if this NO doesn't react with the target molecule slow, then radical and NO eventually react bimolecularly, go back to the ground state. So this RRE is only useful when this free NO react with the target molecule faster than this radical. We repeat this experiment not just the cis-RRE, we also did the neg-RRE and neb-RRE. And tabulated this whole result here, cis-RRE, neg, and neb-RRE. First thing we did measure the thermal lifetime. Cis-RRE has about 2 days lifetime. That's good enough to be useful as an NO transporter, but neg and neb-RRE has even longer lifetime, which is about 5 times longer. So that's useful. Another characteristics, which is similar between cis, neg, and neb-RRE, was the primary NO dissociation quantum yield was about 20 to 50%, which is excitation wavelengths dependent. And those are typically about same to neg and neb-RRE. But after primary quantum yield, the useful NO quantum yield is even reduced to 10 to 30%. But one thing about this excitation wavelengths NO dissociation quantum yields was we wanted to figure out why this is happening. So we took a look at the electronic spectrum of this compound. Basically, electronic spectrums are very similar. They can be composed maybe about 3 to 4 band. And 3 to 4 band, you may think it approaches to about 3 to 4 electronic transition. But when we did the simple calculation, what experimental physical chemists to do, even a simplest RRE substitute to the methyl thiol, you can see very congested electronic transition. And neg and neb-RRE, of course, even more congested, but similar. I can reproduce, observe the electronic transition. And here, with the methyl RRE, we did for strong electronic transition, we optimized molecular structure and found some of the electronic transition lead to electronically excited state. Some of the electronic transition lead to the radical. Even though that calculation is very crude, what it really tells us is the electronic transition is highly congested. It's almost impossible to excite to particular electronic state just undergoing radical. So it's probably intrinsically impossible to get a photodissociation quantum yield of 1 for this size of the molecule. Okay, so probably we better go to the smaller molecules. So we studied photodissociation dynamics of NO from S-nitrous molecule called RSNO, using again femtosecond time of the infrared spectroscopy. The simplest RSNO is again here methyl SNO. And here again, adjusting this the alkyl groups, we can change the characteristics of RSNO, the stabilities, toxicities, and solubilities. So again, we used this biocompatible thiol molecules, cysteine, and its derivative. And these molecules, in fact, found in our body. So that should be non-toxic in our body. And this, using this, we can synthesize RSNO called cis SNO, NAG SNO, and NAB SNO. In the process of investigating these molecules, we found it's probably easier to using thioglycolic acid, mimicking this cysteine SNO, but simpler molecule in the analyzing data, also in the calculation. This molecule have acetate group, but doesn't have ammonium here, and even shorter carbon frame. And we measure the electronic spectrum of these RSNOs, and we found electronic transitions are basically identical. All of them has about intense single absorption band around 340 nanometer. But vibrational spectrum rather more complicated. But again, TGSNO is the simplest. You can see around 1500 wave number shows broad NO stretching mode. And there's also strong CO stretching mode, anti-symmetric, symmetric, and cysteine, cis-SNO having additional ammonium band here. And the NAG and NAB SNO has additional amide band and the CN stretching mode. So to analyze data, we studied the TGSNO. So we excite TGSNO using 330 nanometer obtained time reserved infrared spectra starting from 300 femtosecond again to 10 microsecond. And here you can see immediately after photoexcitation, bleach signal 1600, 1500, and about 1350. And this band is from NO stretching, this one from to CO stretching mode. The bleach appears immediately, so some chemistry going on. But the bleach signal slowly decreases up to 1 nanosecond. But you can see new characteristics. The bleach signal increases again. So this is unusual. And there's a new absorption band appears immediately, but that also decays. Another interesting feature is not quite visible, but here, if you look at carefully, there's a very narrow band, strong, about 1830 wave number. There's a very strong band appears with a few picosecond time constant and decays within a nanosecond. In fact, when we substitute this NO with the N15O, it shows isotope shift. So this band must arise from NO band, NO stretching mode. Anyway, these spectral features all reproduced by basis spectrum adjusting amplitude. And the basis spectrum we used was again ground state TGSNO in the ground state. So CO stretching mode, NO stretching mode, CO stretching mode. And the low line electronically excited state, here you can see CO stretching mode is about the same place, but NO stretching mode here about 1500 wave number shift to the 1800 wave number. That is really dramatic shift in vibrational frequency. And when you look at the geometry of the molecule, in the ground state, this CSNO is in plane, but this electronically excited state case, CSNO, this NO is dihedral angle is about 90 degrees. Not just that, the SN bond length increases. So in the ground state, this NO has NO minus character. That's why the frequency is really lower wavelengths. In excited state, this vibrational frequency shift almost close to the radical character. So the bond is weakened. This got into the radical character. And another basic spectrum is TGS without NO. So this is radical. So there's no NO stretching mode, only CO stretching mode. This is anti-symmetric CO stretching mode. Another CO stretching mode here is outside of this window. And another basic spectrum is coming from this compound with the disulfide bond, have two CO stretching modes. This compound, we synthesized it and measured it. This is not just calculation. So again, by adjusting amplitude of all these basis spectrum, we can get a time-dependent amplitude changes. Here, you can see again, the really bleach is decreases and increases again. And here you see the compound of with the disulfide bond increase around this area. So from this time-dependent amplitude changes, we could get a photolysis kinetics of TGSNO in water molecule, in water solution. So when we excite TGSNO with a 330 nanometer equal to the electronically excited state immediately, about 20% of them goes to here. The excited electronic state decays within a nanosecond to the ground state. About 80% of them undergoes photodissociation, but this, again, this radical and NO is within a solvent cage, and that undergoes Jammer recombination. So most of them go back to the ground state. But another interesting thing is when we photolyze these molecules, we only photolyze a fraction of the molecule we put in, meaning in the environment, there are many reactants still hovering around here. Therefore, the reactant molecule can react with this radical. So that's what really happening. The reactant molecule react with the radical, form disulfide bond, generating extra NO. So due to this reaction, basically you double the free NO molecule. The quantity of free NO molecule is being doubled. And also you remove the radical. So therefore, this free radical can stay here for longer time. So any target molecule reacting with the free NO for a slower time scale, and this RSNO is a good NO precursor, NO transporter. So we repeat this experiment also not just TGSNO, CSSNO, NAGSNO, and NABSNO, and we tabulate the result of them. CSNO turns out to be has a short lifetime, about 5 hours. So probably you can use it, or maybe too short of a lifetime, but NAGSNO has a long enough lifetime, can be used. Now here, interestingly, the primary NO dissociation quantum yields can be up to 100% can be dissociated. But again, whatever the primary quantum yield, due to the very efficient geometry recombination, only 30 to 60% of them are useful for anode reaction. And even this 30 to 60% is achieved because reaction between radical and reactant is doubled free NO. In the process of this analysis of data, we encountered this the excited electronic state NO spectrum is really unique in a sense. Ground state about 1500 wave number shift to the 1800 wave number. So in calculation of this excited electronic state, we had a really difficulty because in fact all these molecules are not a neutral molecule, it is an ionic molecule. And calculating excited states of an ionic molecule was quite difficult for at least for experimental chemists. So we thought maybe a neutral molecule is probably better, but neutral molecule, this MCSNO, turns out electronic spectrum are basically similar, but the vibrational spectrum shows only NO stretching mode. There's no other noticeable vibrational band. So it is very hard to follow any radical and molecule with a disulfide bond. So that was difficult because only NO bleaching and new NO is showing up. So the basis spectrum, we can follow only 2 of them. So it's very limited in a sense. So you can see kinetics is very limited, still get a rough kinetics of them. So we thought maybe we'll just, even though we don't have a data, we'll just calculate excited electronic state of CH3SNO. So this is hardest calculation for experimental chemists, but anyway, we tried to do it and we did it. And the excited state potential, basically we excite to the S2 state. You can see when we excite to the S2 state, you can see some of them go to the dissociation channel, some of them go to the dihedral angle change. So it shows behavior of it, but what we like to have is putting experimentally feasible molecules. CH3SN is extremely unstable. It's very hard to do any experiment with this. Okay, with this, I'll say we got a detailed photoexcitation dynamics of RRE and RSNO, and those are the information we got. And since there are many theoretical chemists, we like to have a better calculation for the excited electronic states. So probably excited state potential for RRE or molecular structure for the excited electronic state of anionic RSNO. Conclusion is basically combining with the well-characterized anode donating precursor, we can probably use photon as a good switch, as anode releasing switch. Okay, I like to finish by acknowledging a student who did this experiment, Dr. Sangchul Bog and Mrs. Jiang Xin, and Ho Jung Yoon did the experiment. And in the calculation, we got help from Dr. Changwon Yang and Professor Youngsang Park at PNU. Also, we got some help from Professor Seung-Gyu Min and his students at UNIST about RRE calculation, which I haven't shown you here. We got a financial support from National Research Foundation through BRL and co-research. And with that, I'd like to thank you for your attention, and I'll be happy to answer any question you